Hello everyone, this is the YouTube presentation about mutation explained by Kenny, Kimberly, and Matthew. Mutation is any change in the DNA sequence of a cell that can be caused by errors during cell division or exposure to environmental factors. Although mutation is not always dangerous, it can be because this change in the DNA can alter the sequence, causing a change in the DNA which can disrupt normal cell function, leading to various health issues like genetic disorders or cancer. First of all, we have somatic mutation. It is a change in the DNA that happens in a cell of the human body after conception, except in germ cells like sperm or eggs. This means that this occurs after the conception of a cell and is not directly passed on to children because these mutations take place in non-productive body cells and not onto germ cells. During this process, you can get a change in your DNA sequence sporadically without the mutation existing in the person's family. But the good news is that it cannot be passed on to future generations. The second source of natural mutation is by translocation. This is just a genetic change where a piece of one chromosome breaks off and attaches to a different chromosomes, that they are a structure found inside the nucleus of a cell. This occurs when broken chromosomes fragments, rejoin, or rearrange their material incorrectly, causing genetic disorders. There are two examples of relocation, and they are reciprocal and non-reciprocal. A reciprocal translocation occurs when two chromosomes exchange pieces of genetic material with each other, while a non-reciprocal translocation happens when a piece of one chromosome breaks off and attaches to a different chromosome without any reciprocal exchange. Now, I will ask you 3 questions and you have to choose the correct answer. Question 1. Which is correct about somatic mutations? Correct is C. Somatic mutations occur after conception, but they are not passed on to children because they are not caused by sperm or eggs. They just occur after conception randomly. Question 2. What type of translocation of mutations is more dangerous for us? Correct is B. Non-reciprocals are the most dangerous due to the net gain or loss of genetic material for not receiving any exchange of chromosomes. And question 3. What statement is false about mutations? Correct is A. Mutation can change the organism's DNA without having negative effects. These neutral mutations are like weak earthquakes that a lot of earthquakes happen every day, but they are not felt because they are not strong.
Now on to mutation sources from external factors. These types of mutations can occur randomly in our bodies. However, mutation can be caused by external factors that if we take care of our health better, we can reduce the chances of deadly mutations in our bodies. The first one is by chemicals. They are called chemical mutations. Chemical mutations occur when a chemical substance interacts with DNA, altering its structure and causing a change in the genetic code, potentially leading to a new trait or abnormality in an organism. These chemical substances are called mutagens, and they appear in our body when we are exposed to X-ray or ultraviolet lights. These chemicals can also be found in tobacco products or even in some food additives. The second one is by oxidation. Oxidation is a common chemical process that happens when atoms lose electrons. Oxidation of cells is naturally occurring in the body, but it can increase by environmental pollutants, certain drugs, cigarettes, or very normal activities such as cooking smoked meat. And finally, the last one is radiation. A mutation caused by radiation is when we're exposed to ionizing radiation. Ionizing radiation is similar to nuclear radiation and is so dangerous because it breaks electrons, damaging our genetic materials in cells. This radiation is so strong that it can be passed to future generations if the damage occurs in the reproductive cells. Alright everyone, it's time for a quick fun game. It's called Fact or Cap, where we'll test your skills in spotting the truth or calling out if it's just straight cap. Chemical mutations are only obtained by X-rays or ultraviolet light. Cap. We can get chemical mutations by things that are not made by radiation, even by things that we consume every day, like additives of common food. Radiations are so dangerous that they can be passed to children if affected by parents. This is a fact. Radiation and radioactivity are one of the most dangerous substances and forms of energy a person can be in contact with, causing blood, skin, and organ tissues. Hopefully you guys learned so much so far. Mutations can occur by factors that we cannot control or even by outside factors by activities we do on a daily basis. Here are a couple of tips and tricks on how we can prevent these. For now, let's go ahead and watch a video.
What does this mean for us? Are we simply at the mercy of time to accumulate mutations until we inevitably fall apart from the inside? Well, kinda, but our body has methods of preventing this. Evolution has learned that having proteins around that self-immolate a cell is a better solution than trying to fix a bunch of problems with the DNA. If there are a lot of problems with the DNA, some of the proteins responsible for fixing it will wake up anti-tumor proteins. These proteins check if the DNA problems can be solved or if they should just start destroying the cell. ATM kinase is involved in repairing extensive DNA damage and also phosphorylates the p53 protein. If there are many DNA problems, then there are more ATM kinases to fix them, which stimulate more p53 protein. p53 is the key protein to solve and prevent cancers, hence its name, tumor protein 53. If there are a lot of phosphorylated p53 floating around, the cell will shut down in a process called apoptosis, preventing potential cancers. It should come as no surprise then that mutations to TP53 are found in essentially all cancers. Let me be clear, not all cancers are caused by a mutation of TP53, but a mutation of TP53 is almost certainly going to cause cancer. This gene is not the only defense our cells have against cancer. There are others. But strangely, it seems it's much more susceptible to mutation. We have 2 copies of every gene. For most of the tumor-suppressing genes, a mutation in one will not disrupt the other. But, in what appears to still not be a fully understood mechanism, a mutation in a single TP53 gene silences or disrupts the functioning of the other. This makes TP53 the key gene to know when it comes to discussing cancer and a cell's defense against them. Swapping one of the 393 amino acids in the p53 protein for another can disrupt the protein's function, preventing it from doing its job. That's cancer. A gradual, continual buildup of mutations in our cells until one of them just so happens to be in the right place to either overwhelm or bypass our cells' natural mechanisms to prevent them.
Real-world examples and case studies. Cancer mutations, when cells go rogue. The first example we have is the BRCA1 and BRCA2 mutations. They increase the risk of breast and ovarian cancer and are passed down genetically in families. Next is the p53 gene mutation. It prevents damaged cells from dying, leading to uncontrolled tumor growth. Next up is lung cancer mutations, or EGFR, KRAS. They are often linked to smoking and environmental exposure. A real-world example of this is how Angelina Jolie underwent preventative surgery after discovering she carried the BRCA1 mutation. Next up, we have genetic disorders, when mutations cause disease. The first example is cystic fibrosis. It is a mutation in the CFTR gene that causes thick mucus buildup in lungs and digestive tract. Next is sickle cell anemia. A single base mutation changes red blood cell shape, leading to oxygen transport issues. Lastly is Huntington's disease. A genetic mutation leads to nerve cell damage, causing movement and cognitive decline. Did you know some genetic disorders can now be detected before birth through genetic screening? Next up, we have mutations in viruses, rapid evolution. COVID-19 variants, Alpha, Delta, Omicron. Mutations in spike protein change ineffectivity and vaccine effectiveness. Our next example is HIV mutations. They evolve quickly, making vaccine development challenging and almost impossible. Lastly, flu virus mutations. Annual flu shots are needed due to rapid genetic changes in the virus. Question: why do you think some viruses mutate faster than others? Next up, we have a real-world example of the COVID-19 mutation timeline. December 2019, original SARS-CoV-2 virus emerges in Wuhan, China. September 2020, the Alpha variant detected in the UK, increasing transmission rates. May 2021, Delta variant spreads rapidly, becoming dominant worldwide. November 2021, Omicron variant identified with higher mutation rates in spike protein. As of today, new subvariants continue to emerge, influencing vaccine updates. Mutation detection and repair, fixing DNA errors. DNA damage detection, errors from replication, UV radiation, chemicals, and stress. Proteins like DNA polymerase and mismatch repair enzymes detect abnormalities. Repair mechanisms, proofreading by DNA polymerase during replication. Next up is mismatch repair, otherwise known as MMR. It fixes base pair mismatches. Next is base excision repair, otherwise known as BER. It replaces damaged bases. Up next is nucleotide excision repair, NER. It fixes UV-induced lesions. Next up is double-strand break repair, where there's two categories: homologous recombination for accurate repair, or non-homologous end joining for quick repair, but it may be error-prone. The importance: it prevents mutation accumulation and reduces cancer risk, and it maintains genomic stability and cell function. Should CRISPR be used to edit embryos? Thank you for listening to our presentation about genetic mutation. We hope you enjoyed.